So I am going to talk mostly about shRNA screening. I'm at the Broad Institute of MIT and Harvard, and at the Broad Institute, historically we've done a lot of sequencing work and looked a lot at structural genomics. So there are various gigantic projects looking at HapMap, copy number, doing GWAS association studies, more recently epigenomic mapping, and a great deal of RNA sequencing. And that yields a lot of knowledge about the structure of the genome in both normal and cancer cell types and other disease states. But what we're interested here for all of us are what's the functional annotation of the genome, and can we use RNAi to actually take what we've learned about the genome and translate that into what is happening in the cell. And so, of course, with RNAi, we can systematically knock down gene by gene and then assess the phenotype. And here are just two example outputs. One is image-based, which Hakeem talked about a little bit already, and the other is expression-based. So I'll mention it a little bit. We have several screens looking at expression data, either in low throughput with a few qPCR assays, or in higher throughput with gene expression, HTS, Luminex, or NASH. So it sounds great to be able to do systematic RNAi, but everyone in this room knows it's not that easy. There are a lot of considerations when you want to do high throughput cell-based screening. First of all, you need an actual tissue culture cell model. So of course you can do whole genome RNAi in organisms like C. elegans, but for mammalian systems, that's not possible for a screening platform. And so you need a tissue culture model that's relevant to your actual biology of interest. And then when you're going to do RNAi, you need to figure out what your delivery method is going to be. Is it going to be siRNAs or shRNAs? What's the efficiency in your model, and what are the side effects of delivery? Hakeem already talked about that quite a bit. All the things we add to make delivery work, transfection reagents, polybrine, pyridomycin, all have effects on the cells in and of themselves. Of course, RNAi is not a perfect technology, and there are differences in different constructs and different platforms in gene knockdown effectiveness. And as we were just talking about, specificity issues, off-target effects, and non-specific effects like interferon effects. And at the end of the day, we have tons of data and a big bottleneck for follow-up. And so there are a limited number of genes we can actually follow up biologically, a limited number of postdocs or graduate students that any given lab is going to send after those genes. And so the analysis of RNAi screens to identify high likelihood, high confidence targets to follow up is an issue. And if you pick the wrong gene, as Hakeem mentioned, your postdocs or grad students get very sad, to say the least, when they spend years